welcome to Beck. And today our speaker is Michael Greenfield, and I actually know Michael from, again, this Center Ennis in France, in Saint-Étienne, France. That's where I first met him, even though he actually was a professor at UCLA years ago in bio. But so Michael is a biologist, and got your PhD at University of Wisconsin, right? And then had then I'm not sure about between there and UCLA, but you came to UCLA and then you went to Kansas and you've been there for quite a while, and then also at University of San Etienne. And I've known about Michael's work for a long time, and my interest with entrainment and coordinated action, and so I've known about this really cool work, and so I'm really excited to hear him talk about it now. So welcome.
Thank you. Well, as Greg mentioned, I had been here, and it's a special treat to come back to UCLA. I was able to find my way around the campus. I haven't been on the campus since 1995. Wow. Yeah, after leaving UCLA, I realized what a special institution UCLA is, and you're very lucky to be at it, and this center. So, the seminar that I'm going to present, it is about time. It's about time, repetition, and most of all about synchrony or anti-synchrony, different coordination of repeated phenomena, rhythm. Okay, rather than give a long verbal description with definitions of what these things are, after this slide I'm going to jump into some animations, animation, and I give this introductory note because there was a very good semi-popular book on synchrony in the universe, astronomy, physics, biology, human affairs. People interested in the stock market are quite interested in synchrony and prediction in time, and sometimes they have contacted me about things. So this book by an applied mathematician, Stephen Strogatz at Cornell, who used to write an occasional column about math for the masses, for the grand public, in the New York Times. It's a good book, covers a lot of different fields, not just fireflies. Okay, so here we go, an animation around the world in sync to give you an idea about the things I'll talk about for the first 3/4 of this seminar. Okay, we've got an oscillogram, 5 frogs singing in a little tiny pond in Panama, Entrainment, 5 frogs. I only had one microphone, so it sounds like cacophony, just noise. But let's look at this oscillogram, amplitude versus time, where I was able to separate out the 5 males because there was a little bit of a vocal signature for each one, and they each had a little bit different frequency profile. So they're called A, B, C, D, and E. And you're going to see the same thing replayed for the same 10 seconds. So frogs have rules. We come back to this later. This is just to get you used to what goes on in the living world in terms of rhythm and rhythm coordination at a behavioral level, because there are other rhythms, of course, in the living world. Okay, so here we go to other frogs, also from Central America, and anti-synchrony, counter-singing. I think you have the idea. Okay, we go on. The counterpart among arthropods, these are, in Europe we call them bush crickets. Here you use the American name katydid, and these are katydids from Panama, and they're going a lot faster than the frogs, so I don't have it highlighted every time they sing. There are two males, the left channel, the right channel, and you've got 11 seconds, and it's a high frequency, so it's not all that loud. And when they do sing at the same time, they're right on the mark. Right on the mark. But they drop out now and then and come back in sync. Okay, another North American arthropod, insect, periodical cicadas. And we know about periodical cicadas. They come out either every 13 years in the south, every 17 years in the north. So you have that very long periodicity and synchrony. But one of the species shows this behavioral synchrony. The males sing. It's a mating call, courtship call to females. And again, this is constrained a little because I only had one microphone. There were literally hundreds of males in the area of an emergence way back in 1998 of the species Cassini. It reaches a crescendo, dies down. 5 seconds later, comes up again, dies down. And you're listening to hundreds of males. Now, it doesn't die all the way to zero, largely because there are males maybe 100, 200 meters away, and they're synchronizing with their nearest neighbors. And the ones near me are synchronizing with their nearest neighbors. So it never goes to silence because they're a different group. So there's a spatial component as well as a temporal component here. But this is, again, if you would have only 2 males, they would be on the mark every 5 seconds. We move over to light, bioluminescence. The same thing. This was on the cover of Scientific American about 30 years ago. These are one of the Southeast Asian synchronizing species. And this species, well, I don't think you really have fireflies on the West Coast. In eastern North America, you have a lot that fly around and flash while they're flying, and the females on the ground may flash in response, and there's a courtship interaction. Here the males are all in a tree, and they flash all at once. I have a film taken from the vicinity of Bangkok. Can you see that, or do we need to dim the lights? Okay, we can go back. Just temporarily. It will. I'll do it again. Okay. The rhythm is about every 600 milliseconds, 1.8 hertz. These are incredibly precise in how they do it. You may notice a little bit of a wave action from one side to the other. Again, the individuals synchronize with their nearest neighbors, and that will move across the population from one side to the other. And you'll see it here again. So, yeah. So we can get that back on. So that's bioluminescence. Still in the realm of visual signaling, but the animals are using reflected light. And this time, other arthropods, fiddler crabs, and this was taken on the coast of Australia, and they synchronize, waving, the males wave an enlarged claw. It's part of a courtship display. There are 3 males here surrounding a central female, and they're all courting her. It's almost eerie. How do they do it and why do they do it? This is visual. Now, as an aside, we know if we're going to do this with acoustic organisms, living organisms that are using acoustic signals, and we start to do experiments, we use playback with loudspeakers. What do you do with visual signals? So this team, I met them in Panama, and we talked about you have to make arrangements with some engineers and build mechanical fiddler crabs. And there are 4 here, and there's a central female, and the mechanical fiddler crabs are timed, and the female has left her place, and she's making a decision based on timing and other things. There she goes. So who knows why, but it was part of a long study. And the final thing, here I mentioned around the world in sync. We're now in the Sultanate of Oman, at the southeast corner of the Arabian Peninsula. And there are crabs on the beach here. They're ghost crabs, and they will rap with a leg on the wet sand, wet, not dry. And there's a recording of 2 males doing this. Now, I'm presenting it to you with sound because we can respond to that. I am fairly certain, although the critical experiments have not been done with ghost crabs, I'm fairly certain that biologically, the biologically relevant signal is substrate vibration, but you can pick that up as an experimenter with the microphone. But the animals, I don't think, have a sensitivity to airborne sound. But the airborne sound is a byproduct of the vibration signal. So these are some things. Okay, we're going to skip across phyla now, and we're going to think about humans. And we're going to think particularly about music, not language. And that's a critical point that we come back to later. So I picked this thing out, Mussorgsky, Night on Bald Mountain. Okay, it'll start. Pay attention to the strings. You have to look at the strings. Okay, now they have a conductor. The conductor helps. And these guys, they're pretty good. They're trained musicians. They're pretty good. It turns out these guys are better, and they don't have a conductor. Some ethnomusicologists studied the precision of human musicians in different cultural venues around the world, and the most precise that has been found was djembe ensemble in Mali, West Africa. They are off by maybe 15 milliseconds, plus or minus. And I don't think I need to remind you, but when we are doing things like this, we have to look beyond Western music. And these are, as far as I know, the best. And there was an article on it. Okay. So with that, maybe we could modify this title a little bit of what I'm going to talk about. So with that animation, I'll give you an overview of the types of rhythm coordination we know in living organisms. And I'm going to start with a very low level of precision and a very long period. Circadian rhythms, or you could think of tidal rhythms, monthly or lunar rhythms, annual rhythms, but circadian rhythms. 5 individuals, and when you see a horizontal white bar, there's activity, and day and night, and a number of things could be going on here. Perhaps it's the evening chorus of birds, and they could be set because they have some endogenous free-running rhythm. That can be modified by the astronomical signal, sunset, change in light level, and also they hear each other. So they influence one another, those 3 things. Lots of stuff on circadian rhythm of all types, not just sexual or communication signals or behavior, but in physiology. Go down to a shorter time scale. This time of year you may hear, if you pass a small pond, frogs that sing. It's mating call, and they'll sing in these collective bouts. Usually there's a bout leader. He might be more motivated, and the others respond. They go for a few minutes, they all die out, and they repeat that ad nauseum during the main activity period of a 24-hour cycle. So again, not much precision, but we see that. But what I want to focus on are what people working with rhythms call ultradian rhythm. Much, much shorter at the bottom, where the period might be half a second, a quarter of a second, 1, 2, 3, 4 seconds, something like that, not longer. And I focus on behavioral things, and I focus on, usually it's sexual and social signaling. Why? Somebody asked me that last week, and my answer to you is why focus there? Those sorts of signals tend to be more precise because the receiver has to hear a certain rhythm, so there is selection pressure on the signaler. You got to be precise. Whereas like we have heart rhythms, physiological rhythms, there you don't have the selection pressure on precision there. And you get what we might call a very precise phase angle, phase relationship between the rhythms, activity rhythm, the song or flash of light of neighboring individuals in the group. Okay, so I'll go over some of the early experiments, many which I've done. I'll start with this katydid from central Panama, 2 males, A and B, just a couple of seconds, 2 seconds. And you notice that you have 3 songs, they're right in sync, then male A drops out, then male B drops out, and then they come back in sync. Okay, now if you're going to study that, very simply, take them in the lab, or in some cases, do the work, experimental work in situ, in the field, and start doing playback. And you can generally see that you have the focal animal with a period of T. You come in with a stimulus here, and essentially the animal prolongs its period, T prime, for one and only one period, and then goes back to the basal or the free-running endogenous rhythm. So it's a very simple thing. We call it a phase delay mechanism, and you'll see different versions of that. I'm not saying exactly the same thing in a lot of arthropods and some frogs, something like that. Now, we go on a little bit more. We have to identify 2 phase relationships, 2 phase parameters. One, stimulus phase. Where in the period does the stimulus occur? If it's about midway into the period, the stimulus phase is 180 degrees. And then the other one is a response phase, which is how much lengthening do you get in the modified period proportional to the endogenous rhythm. So if it goes up 50%, the response phase is also 180 degrees. And then you construct what we call a phase response curve, response phase versus or regressed against stimulus phase. And in this case, it's a diagonal because both stimulus and response phase are about the same. 180, 180, 90, 90, 270, 270, something like that. Now, people who work with rhythm in all sorts of situations, it could be in the physical sciences, in the life sciences, physiologists, plant physiologists, or people working with circadian rhythms will often use a phase response curve to describe what the animals are doing when they're interacting or coordinating. So phase response curve, PRC. So here's the animal again. And here's the phase response curve, or PRC, for that katydid. And it's that diagonal line, a regression line, with a lot of points scattered around it. Now, you start to do more experiments, and I'll just mention one. Take the stimulus and lengthen it. Make it a longer stimulus, longer song. And basically, the phase response curve, it just moves up vertically. It moves up vertically. Lengthen it more. Goes up a little more. And keep lengthening it. And it just goes up. And that tells us something about the mechanism, what the animals are doing. It may not be telling us exactly what is happening in its brain, but at least at the peripheral level, at the behavioral level, we could see how it is achieving rhythm coordination. So, now we could do this algebraically, and eventually we do have to have the mathematical model. But I like to start with a geometric model, which is a lot easier for all of us to understand. We think of the endogenous oscillator in the central nervous system as rising to a peak. A short time after that, there's a little arrow here, a short time after that, the actual signal comes out, the behavior. There's always a delay. We call it an effector delay, little t. Meanwhile, the oscillator in the brain goes down to the basal level and it repeats itself. If there is a stimulus from a device or a neighbor, the endogenous oscillator is reset to the basal level, and then it starts again at the end of that stimulus. And we can look at how that is affected depending on where in the cycle that stimulus occurs. And it more or less, this model is more or less in agreement with what we see in a lot of arthropods and some frogs. And we have the phase response curve down here, which you also already saw. And at the bottom, we have the algebraic model. I'm just mentioning that we can get this full model where we take into account stochasticity, because these are not mechanical metronomes, they're living organisms. And we need that model to do some tests. I won't be really treating that other than to say that we have modeled it and then look at the outcome of the model. So essentially we get an imperfect synchrony if we have two animals using that phase delay. The stipulation is they have to have equivalent tempos. The rhythm has to be more or less the same. And if one is slower than the other, the bottom one is slower, it's going to lag behind. You have a 10% difference. And so 10% difference in endogenous rhythm, they're out of sync. So the synchrony is sort of an emergent property. fortuitously occurs if the animals are the same. And if the difference is greater, like 30%, the bottom one, B, has an endogenous rhythm a bit slower, it basically is shut down half the time. It keeps being reset.
So, but they're converging. It's not just that their onset of whatever signal they make happens to be similar, but they converge toward one another, right?
Well, here, in other cases they are. Here, no. It only happens if you have equivalent tempos. And so, because of the phase delay thing, they mutually reset each other. So they start at the same time. They have equivalent tempos. They're going to go at the same time,
right,
one cycle later, and so forth. Right. But as soon as you have animals that are not equivalent, it gets off a little, 10%, not too much, but 30%. The slow one is, yeah, it doesn't work. So it's emergent. But often animals are pretty much the same at a given temperature. These are poikilothermic, ectothermic animals. So if at the same temperature, they may be the same.
And this is probably selection that to gravitate towards a certain rhythm, so they're mostly the same because of selection.
That's a good point. I think, from having done this, often there is selection to go fast.
Okay.
And so the lucky ones are going fast, but there's a limit to how fast you can go, physiological limits. So I think it's that, and we'll get to that later, that there is selection in the system, but it's at a low level, like to go fast. At this group interactive level, I think it's emergent here.
Do they come in one period? They come in?
Oh, yeah, because they are reset, so they'll come in, yeah, that's right. Okay, now we're going to go to some different animals. So at the top two, these are animals very similar to what we looked at already. But there are some animals among frogs and insects that are a bit slower than what we looked at, and they have a phase response curve, a PRC, that is a lot more shallow. They tend to have a free running tempo that's maybe 2-3 seconds long, and here the situation is a bit different. So, So slower animals, they have what's called in neuroscience a post-inhibitory rebound. So here's the stimulus, and the animal was reset and delayed, but not very much. He can come back really fast. Remember, they've got a slower rhythm, like that, a much slower rhythm. And for animals like that, the phase response curve is very shallow. It's not a diagonal. And if you look at two animals with tempo equivalents, then it's countersyncing, antisynchrony, like that. And this is much more likely to occur. They don't have to be exactly the same, because here, if one is a bit slower, they still go, although one is lagging the other. So this is not such a strict emergent phenomenon as synchrony. Well, so this happens with a lot of animals. And the main thing that seems to be related to is their endogenous rhythms are slower. Some species are slower, like things that we looked at. Okay, one thing about alternation, countersyncing, is if you're going to synchronize, you can have 2, you can have 5, you can have 50, like the fireflies. What do you do if you're alternating and under a different sort of phase response curve? We see essentially two things. One, you could call it triphase alternation. Basically, you interact with your nearest neighbor and you ignore more distant ones. And so A alternates with B, C alternates with B, and by default, A synchronizes with C. And you get this interlocking pattern in space of synchrony and alternation. But the other thing we get, particularly in frogs, we saw that in that opening slide, we get N-way alternation, where instead of having 180 degrees, they may be at 120, at 90, and so forth. So you can pack 3 or 4 in, and they'll do it. It's an interesting natural history observation, but that does occur. Okay, so after seeing that, and I've emphasized the synchrony we saw before is an emergent property, and it merges if the animals have equivalent tempos. Does true synchrony exist? And the answer is yes, and those are the more interesting things, particularly as they relate to some human abilities. So I've shown a few cases. I believe the ghost crabs are doing it, the periodical cicadas. This is a different katydid from Europe, from France and Spain, where they are not using a simple phase delay mechanism. They're adjusting their rhythm. If they fall behind, they shorten their signal so they could speed up, or if they are going too fast, they lengthen their signal and can slow down. And they also have a little bit of a phase delay. So here, this guy was behind, so he dropped out here and then came in sync here. When you see the red, that indicates it's the animal. When you don't see the red, it's crosstalk picked up by another microphone, neighboring microphone. So yeah, we do have true synchrony. And I have studied it in some. This isn't the best example, but there's some cases in bush crickets that are incredibly precise, true synchrony. And by true, what we basically do observe is a coordination of both tempo and phase. What we looked at earlier, just phase adjustment, so you come into sync. But here, the animals, as we considered, may have different endogenous rhythms, and they adjust those rhythms. They converge in this sort of thing. So it's coordination of both tempo and phase, and it is something we know from the physical sciences, a coupled oscillator, an oscillator, another one, and they get coupled. And so essentially, the fast one, the black one, gets slower, and the slow one, the red one, gets faster. And they come into sync here. And there's a fairly simple model that describes this. But before I get to that, physical scientists have known about this phenomenon since the 17th century. Christian Huygens, a Dutch polymath, had some fever, and he was lying in bed in some delirious state, and there were 2 pendulum clocks on the wall. And he's looking, and they're in sync. And basically, we know now how that happens. The pendulum influences the wall, that wall influences the other pendulum, and vice versa, and they come into sync. And basically, the 2 pendulums are, say, like a cricket, the sound and the ear, or a firefly, the flash of light and the eye. And what is connecting them here, it's the mechanical attachment to the wall and the wall itself. And he sent an article to the Royal Society, 1665, an odd kind of sympathy. And you can read that, actually. So this is a model, we call it the Kuramoto model, that will describe it, but we've got to enlarge it here.
Wait, can you just go back to that? What was his story? Did he have any story?
That's a very good point. Not as much as what I'm giving you now, but he was he was a physicist and astronomer, but he was the first one to notice this. he suspected it was some mechanical coupling, but nothing like that. But still, he was the first. Yeah. But we can go and check. I don't think it's certainly nothing like this. This comes from a Japanese man, Yoshiko Kuramoto, who was a physical chemist. And this is his landmark book. And he was looking at these phenomena in different fluids and solutions and looking at waves and things like that. And he came up with this, sorry about that. Came up with this model. And basically it says that the change in the rhythm is equal to the endogenous rhythm plus the coupling of the two rhythms, the fast one and the slow one. Simple differential equation, and you get this synchrony being attained. Okay, we can actually look at this. There's a neat thing on the internet, and I have to adjust some of the parameters. I'm going to make it 2. I'm going to make this 3, and I'm going to lower the stochasticity, the noise, and I'm going to reset it. You see what those things are doing? They're coming together.
Not showing on this screen.
Oh, that's too bad.
You have to.
We had the orchestra, and that went from the internet. That was on the internet.
Is it
What?
somewhere else on your page? Or you've been zooming in and out on your page.
Okay. I'll see if I can do that quickly.
Maybe that you see it elsewhere on your page, but we're not seeing your whole.
Yeah, right. I got you. So it should fill up. No. Well,
You escaped out of it though, and then it showed something else for a minute, whatever you did.
okay. So this should get me into it, and I could see it here.
Yeah. You gotta choose this though somehow in the top of the PowerPoint, and then
Choose this, or no, this.
Yeah.
No. I don't want to delay things too much. So if anyone is interested afterwards, we can look right on the computer. I'm sorry about that. It's a very nice demonstration. And all you have to do is change a parameter, this gamma function, from positive to negative, and synchrony becomes antisynchrony. And we do have some frogs that do that. So, now we get to the fireflies, which are among probably the best in the living world in terms of synchrony and precision. And we already saw this. They're off by 10, 15 milliseconds at most, maybe less. How do they do it? So if we look at the classical Kuramoto model, and if you want to see an individual demonstration later, I could show you, it doesn't achieve perfect synchrony. There's always stochasticity. And so how do the fireflies do it? Because there's always some imperfection. Another applied mathematician, now at Pittsburgh, George Bard Ermintrout, he worked on this because he knew people have known about that firefly problem for a while, because the Kuramoto coupled oscillator, it's a rough approximation. The fireflies are a lot better than that little differential equation. And so he is a modeler. He doesn't work with fireflies. And he adjusted the model. He combined the phase delay that we saw early with the tempo adjustment from the Kuramoto model. He modified the Kuramoto model by adding phase delay, and then he could get it. He could get it. So it's a model. But the problem is, no one has tested the fireflies. They live in a part of the world it's hard to do. We don't know the physiology, but he labeled it perfect synchrony. And we see it in some fireflies, some bush crickets, maybe crabs, some frogs in anti-synchrony, and humans. Humans are quite good, but they're not as good as the best bush crickets or fireflies. They are better. Okay. I want to take a few minutes and talk about why they do it. And I'll start with adaptation. Always someone has, they're selected to do it and so forth. So we've got 4 animals. They're broadcasting some social or sexual signal, and they're more or less in sync, more or less. And this might be because they can retain a species-specific rhythm or call envelope that's important for receivers, usually females, but maybe males, to hear or see. People also suggested you could evade predators, or you could be able to detect predators because you have an open window in which to hear or see. Or you might maximize the intensity, the collective intensity of the local group. So how does that work? Here we have on the left synchrony, and the red trace at the bottom is the collective energy coming out. And so you could see the so-called call envelope separated by no call. And also, compared with absence of synchrony, the collective intensity is a lot higher. So there is evidence for this, because I know in some of the animals I worked with, if you present it like this, the female does not respond to any male. She needs to hear, if it's an acoustic animal, a depth of maybe 10 decibels between consecutive signals. And if she doesn't hear that depth in collective amplitude, she can't pick out any individual calls and doesn't respond to anyone. So genuinely cooperative synchrony. And then if we think about anti-synchrony, there are various explanations for that too. Adaptation. Being able to detect and evaluate rivals and so forth. The other side, and this is more difficult for us to wrap our minds around because I think we're programmed. Oh, there's got to be a reason and explanation. It might just happen. It's emergent. And one way in which that can happen, in some animals, both visual and acoustic signalers, receivers, usually females, but they could be males, they prefer a leading signal, or they only hear or see the leading signal, and they don't detect or respond to one that follows it by a few milliseconds. In human neurobiology, we call it a precedence effect, and we are affected by that. As far as I know, not in terms of our mating preferences, but again, I could show an animation later on that. We're affected by that. Now, if you have that leading signal influence, males who signal slightly earlier than others are going to be favored by selection. And probably the best way you could do that is to use a phase delay mechanism. The problem is, if all the males do it, they may all come in sync, but it's better than being late. Or they may go like this also. The alternation arises, it emerges from this timing preference. And so these are incidental byproducts that happen when you have equivalent tempo and the adjustment of signaling is the same. Sometimes you get synchrony, sometimes you get alternation. So here we got an animal, this Katie did, and we're going to give it two different signals on either side of this arena. One is a short signal and one is a long signal, more energy. And like 11 times out of 12, the female will go to the longer signal. No surprise there, no big thing. With that animal, all we have to do is adjust the phase. Before they were alternating. Now we put the short, unpreferred one just before the long one, like this, with a phase angle of minus 24 degrees. And timing overrides energy. The female goes to the short one because the importance of the leading signal is so great. And this next slide basically gives a summary. The female will go to the long signal, except with this phase relationship like this, where the short one comes just a little bit earlier. And you have that in a fair number of animals, insects, frogs, and it can influence these timing things that we see. Can influence timing. So, but it's an emergent property at the collective level. The synchrony per se is not selected for, it just happens, or alternation is not. Okay, the last experimental thing I'm going to talk about is, it's sort of a, I learned a new American English term on this trip. We're going to do a deep dive. I started to hear that everybody was talking about a deep dive. And so we're going to do that. Did male song really co-evolve with female response, that leading preference? And the way you could do that, really, the only way, you have to use a comparative method, a comparative phylogenetic method. So we've got to get an animal where there are a lot of different populations that differ a little bit, and I settled upon a bush cricket in southern France and northeast Spain. There is a leading preference in the females. The populations are genetically isolated. In fact, some of them, I consider that there are different species, 3 different species, and the other populations, they're genetically distinctive, and they can be tested. Now, we're not going to test them with a full-blown phase response curve. We're not going to be able to do 1,000 animals like that. So we need a quick way of assessing animals here in southern France, northeast Spain, in isolated populations with a comparative phylogenetic method. Now, so we picked 17 populations in that part of Europe, down here. And we now have a phylogeny. It's a fairly crude one done from only mitochondrial DNA, mitochondrial COI data. And we have, for example, some animals sing with short songs and other clade. They sing with longer songs, and there are other little differences. But there's a fairly definite genetic and behavioral differences found between these different populations. Okay, so how are we going to measure things in 1,000 animals distributed among 17 populations? So first, for the female perceptual trait, we can give them a leading and a following signal. How far apart can those signals be and still show the female response to the leading signal? So for example, here it goes to 600 milliseconds, and if they're separated more than that, it doesn't matter. The female goes to both of these with equal preference. So we call F the maximum separation at which females ignore the follower. That's the parameter for the receiver, the female. And then we're going to do the same thing for the male and say we gave the male a synthetic signal, and then after that he doesn't sing for a while because any song in here would be an ineffective following signal. And then afterwards he starts singing again. So basically what we want to do is we want to get the minimum length of time after the signal before the male starts signaling again. And here it's 400 milliseconds. That M in the positioned in the sawtooth oscillator model. So we measure M and F for 17 populations, 1,000 animals, and the prediction is if male song has actually co-evolved with female perception, M should be greater than F, because if M is shorter, it's ineffective. But to more precisely define it, M should be approximately F. That way, the male also maximizes his call rate, both things. So M should be approximately F. So here we have, as an example, two populations on either side of a pass, the Col de Puymarance in the Pyrenees. We've got one population or species on the north side, one on the south side, and they're very different. And here, the maximum, the F is 900, the M is about 900. Here, the F is 600, the M is about 600 here. So that's one example, and these populations are 5 kilometers apart. We graph all them out, simple regression. M is approximately F. Now, if you've used the comparative method, you know that there's a chance of artifact in the sampling, and what we want to do is remove what we call a phylogenetic signal. And the standard method for doing that is phylogenetic independent contrasts. So we get sort of a working phylogeny. It's actually not a phylogeny, it's a genetic structure of the populations. And using that, we can remove the signal. And then we regress it again, and we still get a pretty good thing. So it does seem as if the male phase delay has co-evolved with that female preference. So, but what you get in terms of the chorus, synchrony or alternation, that's an emergent thing. But at the individual behavioral level, yes, it has been selected for. Now at the end, we're going to continue with phylogeny, but look at a bigger picture. And we talked a lot about bush crickets. That's the family Tetagoniidae in the insects. And we have a lot of cases where there is rhythm coordination, synchrony S, alternation A, and the phylogeny is huge. So this part on the right is appended below this. That's the whole thing over here. And if we look at the distribution of these known confirmed cases of synchrony or alternation, there's no apparent pattern. They're scattered around. And even different kinds of synchrony. True synchrony, emergent synchrony, it's all over the phylogeny. So that's something to remember. Now let's look at a bigger picture. All multicellular animals, metazoa. And the only cases where we see these sorts of phenomena are in arthropods, an example there, and the vertebrates and the chordates, vertebrates. And there's a human over there. So that's where it occurs. That's who entrains. And we're talking about a separation, a most recent common ancestor of at least 550 million years, at least. And it may be a lot more because some people think the separation goes way back before the Precambrian, maybe 800 million years. And yet we see such remarkable convergence. So it's undoubtedly an analogous thing, not homologous. Okay, and that's where we come now to the end, us humans. because as far as we know, humans are the only animals other than some frogs and these various arthropods that can do these things, particularly the accurate synchrony. And I'll mention that I've been studying these things since 1980, a neat natural history phenomenon. Beginning around the year 2000, people working with music and human cognition started contacting me. Could we see these models with the katydids? Do they apply to us? And more than that, could they apply to non-human vertebrates like cockatoos, sea lions, different primates, and so forth? Well, we've had a few recent meetings in Europe, the last one 2 years ago in southern Sweden, and I got to meet this fellow, Bjorn Merker. He's long since retired. He's a polymath, like you could say Christian Huygens, and he is an MIT-trained neuroscientist. He got into it because as a jazz musician, he wanted to learn how we perceive and make music. And he got into it more and more. He's interested in the brain, he's interested in music, and he's interested in these non-human animals that can do it, including katydids and frogs and fireflies. And we analyzed a lot of the recent claims. Oh, there are some parrots and sea lions. There is no synchrony there that would in any way approach what I showed earlier. It is, and the other thing is, all of those cases involve animals that were trained by a gifted human trainer for a few years, or they lived with humans for a long time. And even then, when you analyze what they do, it's not what the orchestra does or what the fireflies or the katydids do. It's far less than that. So, perfect synchrony in humans. And we see that in dance, drill, clapping, chanting, as well as vocal and instrumentation music. It's these things. One last empirical slide. Humans are typically tested for this ability in cognitive labs and psychology departments. And a very simple tapping test will reveal the ability. You have a metronome, and it's going at a certain rate, and you're asked to tap along with it. The controller turns the metronome off, and the experimenter measures the continuation of the rhythm. And this is what you see. These are different rhythms that are tested, and this is why the ordinate indicates how far off you are. 20 milliseconds out of 600 or 700 milliseconds. That 20 millisecond error, deviation, is less than our effector delay between the brain and the motor activity. So yeah, we're pretty good, pretty damn good. And this was done with people who had no musical training or no special musical training. On the right, we go one step further. You have the metronome, and you have the human, and you say, okay, tap along with the metronome, and then the metronome still keeps going. And how close are you to it? And these are the different metronome rhythms. And this will show you here on the right how far off you are. And you're talking about being off by about 10, 15 milliseconds. Almost as good as the fireflies, but not quite. Or the same with some bush crickets. So again, we're pretty good. So a few more things. Precision. The mechanism. People have studied the mechanism in humans. It is a combination of phase adjustment and tempo adjustment, like animals, non-human animals, arthropods. You don't need any training for it. Now, does musical training improve it? I don't know. Bjorn Merker probably would have an idea. He is among the world experts on this. Ontogeny, and this is the interesting thing. People have studied it in infants, and we know infants can respond to rhythm, recognize it. Can they synchronize it? That ability to synchronize basically is only developed at puberty, from what I've read. So it makes you wonder about the role, about the sexual selection origin in these abilities. And we know from the animals it's all tied up with that. So the interesting thing here I'll mention, it's not found in natural populations of non-human mammals or in birds. So you may know about a famous cockatoo named Snowball. You could see him on YouTube. First of all, if you really measure what Snowball does, it's not synchrony. But the other thing is, no parrot in the wild does anything like that. And as far as I know, there are no primates that do anything, no non-human primates that do something like this. Some of them can be trained a little in the lab, but not the synchrony. And so we infer some early hominid origin 4 or 5 million years ago. We have to get more information on bonobos because there's a lot more knowledge on chimpanzees than bonobos. But there was a little article written by Frans de Waal, who passed away very recently, that he wrote 35 years ago suggesting he saw it in bonobos. But nothing since then. Nothing since. Early origin. But a real important thing to realize here, and I've had people working in human communication departments. Well, it's in human language. No, sorry. You don't have this kind of rhythm coordination in human language. We may have rules for conversation, which are like duetting in birds. But this, no. This is music, military drill, dance, clapping, chanting, things like that. And so we wonder, where does it come from, this thing that is a major component of our musicality? Because once you get beyond a solo, whether it's vocalization or instrumental, you're going to be timing. And except for we have cases of counter singing and so forth, but generally it's in rhythm, it's in sync. And where does that come from? How did it arise 5 million years ago? I have some ideas, but Bjorn does with me, but I guess I'll leave it go at that. And we could talk about those ideas. And I also, if you're interested, I could show these little animations. But I've gone long enough, so I'm going to stop. So.